We're P4G5, and this is our excitation, contraction, and coupling and sliding filament theory video. And I'm Victoria Wittenbrock, 4.33.
I'm Ben Milton, 4.16.
I'm Noah Villanueva, 4.31.
So, starting with the muscle we have here, the muscle is attached to the bone using tendon. The muscle tissue is shown here by this yellow paper. That is the outermost portion of the muscle. It is covered in the epimysium. Within the muscle tissue, we have this blue paper, which are fascicles. Each fascicle is covered with paramysium, and within each fascicle, we have these black portions, which are muscle fibers, and they are covered in the endomysium. And within each muscle fiber, there are myofibrils, which are covered in the sarcolemma. And within the myofibrils are myofilaments such as myosin and actin.
So this is the sarcomere, which would be in the myofibril. And this is what exhibits the contraction with the sliding filament theory, is when it comes together. So this would be the Z disc, which this is the actin, and it's bound to the Z disc. And the area that has just the Z disc, the actin that's bound to the Z disc, is the I band. And then this is the myosin, which is in the middle, and the area with just myosin is the H zone. And then the area where the myosin and the actin overlapped is the A band. So in the sliding filament theory of contraction, when the H zone is minimized and the actin is brought together by the cross bridges of the myosin and it's pulled together, that's what causes a muscle to exhibit contraction.
The excitation contracting coupling starts when the action potential in the motor neuron reaches down all the way to the axon terminal, and once it reaches here, it sends acetylcholine through the synaptic cleft to the acetylcholine receptors. And once the acetylcholine reaches its receptors, it depolarizes the sarcolemma, which then sends a signal through the sarcolemma all the way here down the T-tubules, where the T-tubules will start to open up the passageways in the sarcoplasmic reticulum so that the calcium can go through the passageways into all the way to the sarcomeres in the myofibrils.
So this is a bigger scale of the sarcomere over here of the myosin and actin. So this would be the actin and this would be the myosin, and the beads would represent the myosin binding sites on actin, and the cord would represent the tropomyosin, which blocks them, and then the knots would be the troponin. So then when the calcium or the quarters come in, they bind to the troponin and they rotate it so that it moves out of the way. And it moves the tropomyosin out of the way, and the myosin binding sites on actin are uncovered, and the heads of the cross bridges of the myosin can bind to the myosin binding sites on actin, and then they do a rowing motion. They pull the actin this way, and then more calcium would come in and bind and rotate again and move it out of the way so that the heads of the cross bridges of the myosin can attach and pull it yet again, so then it slides more. And then that's what would be exhibited here as contraction when the myosin pulls the actin closer together. So it's like this.
So the myosin is able to bind to the myosin binding sites on actin because of the presence of ADP and P. Once ADP and P are in the cycle, they are what allow the myosin heads to come across and bind to the myosin binding sites on actin. And then once they've bound and they have pulled the actin, the ADP and P leave the cycle and a new ATP molecule enters, and the ATP is what allows the myosin heads to detach from the myosin binding sites on actin. And once it's completed detaching, the ATP undergoes hydrolysis, which will cause the ATP to be broken down into ADP and P, which will then be back in the cycle and allow the myosin to reattach to the binding sites on actin, and it will basically allow the process of sliding to continue to occur.
And when the muscle is ready to relax, the brain stops sending action potential through the motor neuron, and that causes the axon terminals to stop sending the acetylcholine through the receptors. And when there's no more acetylcholine at the receptors, the sarcolemma polarizes once again, so it stops sending signals to the T-tubules. And once that happens, the sarcoplasmic reticulum starts to reabsorb the calcium, like this. And once the calcium is out of the sarcomere, the troponin rotates back into place so that the tropomyosin can go back to its original spot where it covers the binding sites on the actin, so that the myosin can no longer attach itself to the actin and pull it further. So as you can see here, when it relaxes, the actin the actins go farther apart and the H zone expands back to its place.